Welcome to the RedChip Virtual Conference.
Our next presentation this morning comes from Jim Czir. He is the Executive Chairman and Founder of Galectin Therapeutics. Galectin Therapeutics trades under the symbol GALT on the Nasdaq, and that presentation will begin momentarily. Okay, Jim, the slide deck is up. You may begin when ready.
Okay. Well, I want to thank everybody for attending. Galectin Therapeutics has been located in Newton, Massachusetts, and we are happy to be here today. You are all familiar with forward-looking statements, so I'll move past that, and let's talk about some of the investment highlights of our company. First of all, we have first-in-class proprietary compounds that inhibit galectins. This is a new area of scientific endeavor, and has been proven that galectins are heavily involved in many different types of disease pathologies. We're using complex carbohydrate drugs, which have a very safe profile. We have two compound categories, the GMCT class, as well as our GRMDO2 class. We've got preclinical models that show that galectins are critical targets for diseases. They're both very, very large market opportunities. One of them is a cancer drug, and one of them is a drug for liver cirrhosis. Both very, very big. We have intellectual property that's our, invented in-house. We've got methods of patents granted. We're looking at our liver fibrosis patents, which have just been issued, and we have a couple other ones pending. A very experienced management team. We'll talk about them in great detail. And I'm having a hard time with the advancing the screens. There we go. Peter Traber is our CEO, and a gentleman that I'd like you to spend a lot of time with. Dr. Traber previously was the Chief Medical Officer in charge of global drug development at GlaxoSmithKline, as well as being the CEO of both University of Pennsylvania and Baylor College of Medicine. If you check out Peter's bio and background, you'll notice that, and compare the SEC filings with our company, you'll notice that he has taken approximately $1 million lower salary than what he's accustomed to in order to take this job. And I'll show you some slides that will give you an indication about why he's so confident in what we're doing. The other gentleman I want to mention is Harold Schlevin. Schlevin has just joined the team. Had an announcement about that earlier this week. He's got 25 years experience in senior management in the drug development business. He's been CEO of Solvay Pharmaceuticals. He's also been very instrumental in a lot of developments like, starting the SIVA Vision Ophthalmic division for SIVA. Milestones the company's looking at. We are hosting a global conference on galectins in disease in a couple weeks in Boston. People will be attending from all over the world. We'll be getting a lot of publicity about that, all designed to study and share information that's been researched about the role galectins are playing in disease. In November, we are presenting at the American Association for Study of Liver Disease abstracts regarding our NASH findings. In cancer, we have a patient that's already been infused, and we're starting the 2nd through 12th patient in a stage 1, phase 2A melanoma trial. It's a two-stage trial. It's a 2A proof of concept trial, which we'll discuss in greater detail. In March of next year, we believe we'll get top-line results with the first 12 patients. Fibrosis, we've already met with the FDA for our pre-IND meeting. We expect to start a clinical trial for non-alcoholic steatohepatitis or submit in January and start the trial in February, with results next year.
Jim, if I could jump in here, we're still on the investment highlights slide, so I don't believe the slides are advancing.
Okay. What are we going to do about that? I've got a border around my screen that says move this window away from the shared application. What is that? Should I give control to you, Jim?
Yes, please.
Okay. Your mouse. Did you get it yet?
I'm trying here, but let's see. Let's try this one more time here.
Okay. So you have it now?
I just need you to click the Show My Screen function for me.
Click it.
Yes, please.
Okay. Are you good then?
Believe so.
Now, are you in charge of the slides or am I in charge of the slides?
I am in charge, but I will give it back to you.
Let me know when I'm supposed to have it.
Okay.
Okay, so we're back on milestones, which we'll be discussing and reviewing again, so we'll go through these rather quickly and get back into the actual science since we're a little bit behind schedule. So let's talk about the science of galectins for a minute. We're either critical participants in the pathogenesis of many diseases, particularly in cancer and neoplastic diseases like cancer. We're looking at the left-hand side of the screen here. We've got a cell that's secreting galectins that are illustrated by these half moons. They bind to cell surface and matrix glycoproteins. They do a variety of things, but the things that we're most interested in are that they're markedly increased in inflammation, fibrosis, and cancer, and that these galectins promote the pathology of those specific diseases. If we look at our compound, you'll notice on the lower left-hand corner that our molecule is a polymer. It's got multiple binding sites, and it actually binds up the galectins and inhibits them from doing what they would otherwise normally do. These are novel carbohydrate complexes that target galectins. They have a very strong binding affinity to galectin-3, which is the most prominent galectin in the disease process. We believe we're interrupting multiple cellular pathways, which is one of the reasons our results have been so strong. This is an entirely new class of therapeutic agent. Again, very low toxicity. These are made out of carbohydrates that are food substances or things that are used in baking and cooking. I'm sure you've ingested hundreds of pounds of it during the course of your lifetime. We have two classes, the GM series and the GR series, and we have a very low manufacturing cost because you can buy these materials by the shipload for a few dollars a bushel. And so our production costs are very, very low, as well as their safety. If you look at intellectual property, the GM class, we have a variety of patents issued already. We have 14 international patents, 5 U.S. patents, 5 international patents pending. We have a method of use and NASH patent that's pending with priority from 2011. We've got our first liver fibrosis patent was issued here a few weeks ago. Had numerous inquiries from large pharma since then. The GR class is the class that we're taking into the treatment of NASH and liver disease. Composition of matter, as you can see, is pending. The method of use was issued here with a priority of 2006. And we have method of use and NASH patent pending with priority of 2011. It's important to note that all of this intellectual property is generated in-house. We have no encumbrances, no overriding royalties to anyone. Everything has been assigned by the inventors to the company. And something else to consider, because these are carbohydrates and they're larger molecules compared to a lot of drugs, these would be very, very difficult to synthesize, but we don't know of any technology today that would allow you to do that. And so when it comes to the equivalency that generics are always looking for, we believe that that will not be available with this class of compounds, that people will have to go through the entire development program with their preclinical and all their clinical trials to get a drug approved. So let's talk about the immune enhancement in cancer therapy. This is an entirely new category, and we'll get involved in the mechanism of action here in a moment. Galectins are involved in a variety of the activities in cancer. They're involved in angiogenesis, they're involved in adhesion and detachment, invasion and migration. We're going to focus on the tumor immunology today. It turns out that galectins are very, very critical and interfere with the body's T cells. What I'm going to share with you right now is information that we learned from the Ludwig Institute, who had been researching this issue. And what they've found is that tumor cells excrete Galectin-3, and that this Galectin-3 binds to the T cells. You can see on the cartoon caricature here the illustration of the red coating that's being put on the T cells. When this happens, the T cells become, as the biologists say, anergic, or perhaps a more common way to describe it is that they stop recognizing the tumor cells as a problem and they ignore them. They stop producing cytokines that they would normally use to kill the tumor cells. And so they cohabitate the same space and the tumor cells grow unabated. Now this is a recent discovery, and this is a very significant finding that explains why cancer has been so difficult to beat up till now. Now they've also found out at Ludwig Institute that our molecule GMCTO1 blocks this mechanism. It binds to the galectins so that the T cells are not coated, and they continue to produce cytokines and kill tumor cells. What's even more exciting is that the research indicates that cells that have already been deactivated by this galectin effect can be reactivated by our GMCTO1 molecule. So we're excited about the clinical trial that's going on. But before we discuss the clinical trial, let's talk about what we know about this molecule GMCTO1. First of all, we know it's demonstrated safety in over 100 human subjects that have been involved in a phase 1 and some partially completed phase 2 trials. If we look at the first bullet point, we had a phase 1 trial with 40 subjects, and it was considered safe. In the phase 2 trials that we conducted, one of them was a 20-patient trial done by prior management prior to the turnaround of the company. What they found there was they had a 46% improvement in survival from an average of 4.6 months baseline to 6.7 months when treated with GMCTO1. That compares to historical controls where Erbitux had a 6.1 month median survival, so an improvement. When we completed the studies and actually compiled the data from multiple studies and compared the serious adverse events that were associated with 5FU, they were markedly reduced. And when we say markedly reduced, normally you would see between 14 and 40% of the patients having grade 3 or grade 4 diarrhea. In this case, we had 0 of the patients had grade 3 or grade 4 diarrhea. Mucositis was greatly reduced, as well as anemia and nausea. We took that data and went to the FDA, and they actually approved a phase 3 clinical trial with GMCTO1 in 5FU for line 2 metastatic melanoma patients. We elected to do something different, and this is what we elected to do. We elected to collaborate with the Ludwig Institute for a couple of reasons. This Galectin effect is something that every vaccine is going to be confronted with and have to deal with. And when you consider that there are 100 vaccines in development today, then we chose to be an enabling technology for the state-of-the-art in the direction that the oncology treatments are moving, instead of being a way to improve an old-fashioned chemotherapy agent that everybody else wants to obsolete. So what we're looking at here on this slide is the current ongoing proof of concept phase 2 trial that's going on in Belgium. And if I can back up a minute. And what we're looking at here is you can see that the blue arrows on this timeline indicate an injection with the vaccine. So the patient's getting the vaccine every 21 days to build their T cell count. And then every 3 days you can see the red injections illustrating the infusion of our GMCTO1 molecule to reverse the galectin effect or to prohibit the galectin effect in order to keep as many of the T cells fully functioning and killing cancer cells as possible. We've looking forward to the completion of this, we started injecting in May. We're looking for top-line results in the first quarter of 2013. We believe that with favorable top-line results, we've got a couple different options, many of which will involve joint venture opportunities with a variety of drug companies that are working on current vaccines. We could continue the same proof of concept trial we have, or we could abandon it and go into a full phase 2B trial. That may be funded by a drug company, may be funded internally. We can look for partnering opportunities, but certainly in the near future, we're going to have a milestone that could change the character of the company. This immune enhancement by blocking Galectin Effect also appears to be synergistic. We're looking at the ability of the immune system to recognize and kill tumor cells is a very active area in personalized approach to medicine today. And since we inhibit this, the debilitating activity of the immune system, we think we could be very, very important to a lot of different companies. There's already 2 agents in the market. You probably all know about dendritic cell vaccine Provenge and the T-cell activator Yervoy by BMS. But there's an estimated 100 additional vaccines and activators that are in development at the moment. And frankly, we don't really care which one wins. We don't care which ones really work, because we know that whoever the winners are, they're still going to confront the Galectin Effect, and we will be there synergistically to help them. We know this is a very, very large market. It's estimated to be over $7 billion by 2015, and that's just the beginning. We think we're best positioned in this marketplace. We know that there are other competitors. You go down to the third bullet point. We've got some competition. Galecto Biotech is in discovery phase in Sweden, working on small molecules. La Jolla Pharmaceuticals bought a GCS100 compound from Selena Therapeutics. They've got it from Prospect, who got it from Pathogenesis. It's a blood cancer treatment. It does bind to Galectin. It appears to have some significant side effects, reportedly. Mandel Med in Oakland, California hasn't done anything with their truncated Galectin-3 protein. And the Stellic Institute in Japan is in preclinical studies on an RNA approach for inflammatory bowel disease. So having said all that, we think Galectin Therapeutics is best positioned for the human trial on cancer, immunotherapy. And since GM-CTL1 is already proven safe in 100 patients, we think this is a very strong approach and will be viewed favorably. So let's move on to the liver fibrosis for a minute. This is an extremely exciting situation. Liver disease is a big problem. There are only about 6,300 transplants a year. The waiting list is 3 times that. Many people don't get on the waiting list. There are deaths of 44,000 a year from cirrhosis, and these numbers are old. With the cases of hepatitis C burgeoning and the explosion in non-alcoholic steatohepatitis or NASH, it's estimated these numbers are probably conservative. You can see at the bottom there's 9 to 15 million patients estimated to have NASH. That's according to an NIH estimate. They expect that 20% of those patients will ultimately develop cirrhosis of the liver. That would be as many as 3 million additional cirrhosis patients. As you can see, the transplant number of 6,300 is woefully inadequate to solve that problem. So Galectin-3 is a critical protein target for therapy, and why do we know that? What's our key evidence? We know that Galectin-3 is produced in large amounts by a fibrotic liver, and this is both animal and human tissues. And we also know that Galectin-3 is essential in mice for the development of liver disease. They've actually exposed mice to toxins that were missing Galectin-3 gene and were unable to create scar tissue like they do in animals that do have the Galectin-3 gene. We know that Galectin inhibitors that block the production of this fibrogenic marker in key human stellate cells is responsible for fibrosis, and we also know that when we inhibit these galectins that we actually reverse fibrosis in animals. What you're going to look at here is a study that was done. This is the study that convinced Dr. Traber to take our job as CEO, and this is also part of the information that has been responsible for 11 key opinion leaders in the world of liver research to join our clinical trial development team or scientific advisory board, as the case may be. So what you're looking at here is a rat liver that has been created by injecting a normal rat with thioacetamide for 8 weeks, and you can see all the scar tissue riveting like a sponge, through the liver tissue. And this is severe, severe situation. Well, after 4 weeks of treatment with our GRMDO2, the animals that came out of that same group that had been treated for 8 weeks with thioacetamide saw the scar tissue in their livers clear like the animal on the right. This is an amazing phenomenon. If you were in the liver disease research business, you would question whether or not somebody was playing a game with Photoshop, because it's outside of the realm of possibility, according to researchers who have been struggling with this forever. We also then decided to try to recreate NASH in animals, and we wanted to do it without chemicals. We wanted to do it the same way your neighbors get the disease. Typically, they eat too much high-fat food, become type 2 diabetics, and then they continue to eat high-fat food, and they develop non-alcoholic steatohepatitis, or fatty liver disease. So what you can see here, what we did is we took diabetic mice, fed them lots of high-fat foods, and in 5 weeks they had fatty liver disease. It progressed to NASH in 7 and became actually fibrotic livers in 9. What we're looking at here on this slide is the vehicle is an animal that has the disease. It's been created. You can see lots of white nodules. That's fat tissue. On the bottom slide, which is treated with GRMDO2, you see just a very few little specks of fat. The white in the center is a cross section of a vein that's not fat that you're looking at there. If you look at the pathology scores on the side with the GRMDO2 on the far right in blue, you see it has reduced the activity score, which is a combination of fat, inflammation, and ballooning hepatocytes by about 50%. But people don't die from fat in their liver. They die from the resultant scar tissue that grows as a consequence of the inflammation that all this causes. So we decided to measure the scar tissue or the collagen content. And what you're looking at here are 2 different sets of animals that were treated when the disease was relatively early in its formation and when the disease was later in its formation. You can see the animals that are marked in black. Those were part of the control. The animals that were treated with GRMDO2 in blue has brought the scar tissue down. You can see in the very center between the 2 slides, it says normal level, 0.33. You can see that it's in 4 weeks of treatment, it brought the amount of collagen down to normal background levels, something nobody's ever been able to do in a NASH model that we know of. To complete the cycle here, we've also then measured for the presence of Galectin-3, the activator that's responsible or very important in the development of scar tissue or collagen. And you can see that on the left-hand side, the vehicle or the animals with the disease, the Galectin-3 stains brown, and you see an ample amount of it. On the right-hand side, the post-treated with GRMDO2, you see very small background amounts of it, which is fine. And by the way, people ask the question about what happens if you eliminate Galectin-3. Well, these Galectin-3 deficient mice that have had the Galectin-3 gene removed and can't have Galectin-3, they live a normal life. They reproduce. They gain weight. They have this average weight of a mouse. There appears to be no difference. And so by just removing this amount of galectin in the liver, we can reverse the disease without doing any damage to the animals. This slide here is a little bit complicated and we're a little short of time, but I'll take and grab some of the highlights. This metabolic syndrome that starts the cascade of events that gets the scar tissue started can affect a variety of cells, and you can see hepatocytes, bile duct cells, and macrophages. And you also see that there's the stellate cells that are quiescent in the liver, but then they start to become activated, and when they become activated, they start producing an extracellular matrix, and you can see the the picture of the scar tissue that is organized there. There are systems in the liver to metabolize small amounts of scar tissue, but it gets out of control when this process gets started and gets cascading too greatly, and then it's impossible to catch up. A little bit like the guy who's got a mortgage and he's paying interest, and the interest is larger than what he can afford to make a monthly payment on. it eventually consumes him and bankrupts him, but if he can make payments that include some principal, ultimately he can metabolize it, or take care of the loan. Liver can do the same thing with the collagen. What's interesting here, what I wanted to show you, is if you'll notice these different pathways, Galectin-3 is implicated in multiple places. And we think that one of the reasons our drug is working so well is that we block Galectin-3 in all these places. These other moments or information on this slide is a little more scientific, gentlemen. I'm going to leave that for anybody that wants to call and discuss it personally, and because of time. The NASH development timeline is that we've already had our preclinical meeting with the FDA, and we are finishing the preclinical studies that we need to do to submit in January our IND. We expect to start our phase 1 trial in February. We expect to finish and have top-line results in the 4th quarter. This is going to take a little longer than we originally had expected, but that's because the FDA has asked for us to do multiple dosing of patients and not single dosing studies. The upside on that is that the multiple doses of 4 weekly doses should give us some indication of efficacy in this trial from the standpoint of liver functions. So that's an exciting proposition because that could lead to some early collaborations. We expect that we'll finish the phase 2 trial in Q4, and it will start a phase 2 trial in 1st quarter 2014, and then it'll take approximately 12 months to complete. The phase 2 trial is going to have biopsies involved in it as a method of proving reduction in scar tissue. And with that successful endpoint, we expect that we will be receiving offers to be acquired. Examples of different therapeutic approaches. They've tried a variety of things. They've looked at lifestyle and dieting. It can be beneficial, but it doesn't reduce the scar tissue, and it certainly doesn't work in advanced stages of NASH. They've tried to treat the diabetes, and it hasn't worked. They've protease inhibition, that line, 5th line down. They actually had liver toxicity. Cysteamine, the last one in the example section, is a drug that Raptor Pharmaceuticals is testing right now on adolescents with NASH. We think that it's not that likely to work because it only addresses one of the 4 different pathways that we showed you earlier. We think that GRMDO2 is actually well positioned and is best positioned with respect to competition because we've got the most attractive mechanism with multiple sites of action. It's independent of hyperglycemia, and it looks like it should reverse scar tissue or fibrosis, which at the end of the day is the real critical factor in this disease, and it's the factor that causes liver failure. And again, our toxic profile of our compounds are very, very good in that they're carbohydrates. So back to the milestones we missed earlier in the beginning. We've got a conference coming up here later this month, or in September, that we're going to be hosting in Harvard and Boston at the Harvard Club. We are going to be presenting in November at the American Association of Society for Liver Disease, our abstract on NASH. We've got the cancer clinical trials that are ongoing with our stage 1 top-line results expected at the end of the 1st quarter. In fibrosis, we expect to get our IND submitted in January, and we expect to start our phase 1 trial in February and complete it in 2013, and then kicking off the phase 2 trial in 2014. We've got roughly 15.6 million shares outstanding. We had $13.1 million of cash in the bank at the end of the quarter on 6/30. We're burning about $450,000 a month on a historical basis. If you look at the warrants and options and things that are outstanding, there's I want to bring special attention to the preferred Series B, 2 million shares on a converted basis. In the warrant Series B, 5 million shares on an exercise basis. Those are all owned by an entity called the 10X Fund, of which I'm the general partner and operating manager and largest shareholder. And all of those have a lockup provision until May of 2015. So unless somebody dies and needs to liquidate their estate, which we would do through crosses with institutions or hire an investment banker to place, those are locked up for the duration. So with that, I'd be interested in turning this over to any questions.
Thank you, Jim. First off, I'd just like to say thank you for all those listening and for RedChip companies for taking the time to present at the RedChip Small-cap Virtual Conference today, and we will go ahead and jump in to Q&A. First question here. Do you anticipate potentially encountering any roadblocks that could cause Galectin to miss its target of stage 1 top-line results for cancer by March of 2013?
I'm sorry, I couldn't quite hear the question.
Sure, no problem. Do you anticipate potentially encountering any roadblocks that could cause Galectin to miss its target of stage 1 top-line results for cancer by March of 2013?
Do I expect them? The trial is being run by the Ludwig Institute, which gives us great leverage because they're actually paying for it. We're providing a drug, and we think that's a very unusual situation and to tremendous advantage to the Galectin shareholders. As a consequence of that, we are not physically running the trial and in charge of patient recruitment ourselves. So it's possible that number could slip a month or so. On the other hand, because of the type of trial and its design, there's a possibility that news could come out earlier. For example, metastatic melanoma is a type of disease that doesn't give up survivors and takes people down on a very quick basis. If we were to have a couple of the early patients in the trial have a total remission of the disease, that news could be disseminated. And so it's to answer the question, we hope there aren't any delays. We're not in control of the trial ourselves, and so it's a little bit hard to to represent that there couldn't be a delay. We don't see what that would be, though, because the patients are pretty easy to recruit for a disease like this. It has so few things that can be treated with, and the Bristol Myers drug's not approved in Europe where the trial's being done, so it's unless people want to travel to the United States, it's the only it's one of the few options that they have.
Thank you, Jim. Next question here. Just to clarify, your proposed treatment for liver fibrosis would help cure liver fibrosis and prevent the need for a liver transplant. Is this correct?
Well, the idea is that it would reverse the scar tissue. Now, how will it work on extremely late-stage patients? If somebody has gotten to the point that the pressure has built up so much in their liver that the vein that is feeding the liver is leaking, and blood is oozing into areas of the body it's not supposed to, and it's only days away from bursting and the gentleman or woman dying from internal hemorrhaging, it's unlikely that patient's going to be curable. But reversing the fibrosis could very well eliminate the need for many of the liver transplants that are needed today.
Thank you, Jim. Next question. I believe you may have touched upon this in your presentation, but have any side effects from your treatment been seen yet?
No.
Excellent.
No, as a matter of fact, in the product, the GMCT01 that has been used in humans, there have been 100 patients that have had the drug. 57 of them have had the drug at the highest dose level. When we compared the severe adverse events from the people who had 5-FU treatment, those who had 5-FU in our drug, the people who had the chemotherapy in our drug actually had lower incidence of severe adverse events than historical controls where people had no chemotherapy at all. So we do have a significant effect on side effects.
Thank you, Jim. Next question here. What does GALT hope to accomplish by the end of their September conference?
By the end of the September conference, well, we are focusing and very excited about getting our liver trial started in February, and our IND submitted in January. So that is our our primary focus. The purpose of the conference is to continue maintaining our position of leadership. We get a lot of academics that call us and want to collaborate, want to share information with us because they know us and recognize us as the leaders in Galectin therapeutic research. So we're actually publishing a book, and along with having the conference, that'll be the second ever book on using galectins for treatment and for developing treatments for disease. And so this is the conference is there to facilitate the collection of data and the advancement of research, but it's also there to continue to bring world-renowned experts in research into our funnel so we can see what they're doing and perhaps benefit from that and help advance these therapies to marketplace.
Thank you, Jim. Next question here. Is Galectin considering any additional funding over the near to medium term, and if so, what types of funding would you consider?
In the near term, medium term, the only funding that we're going to consider would be grant money, joint venture money, or some additional infusion of capital by the 10X fund that I'm a manager of that would be exercising some warrants, and those would again be subject to the May of 2015 lockup.
Thank you, Jim. Next question here. You mentioned being a possible acquisition target during your presentation. At what trial stage do you think you would need to advance to attract attention for that to occur?
That's a very interesting question, and obviously involves a little bit of guesstimate. Let's take a look at Inhibitex and Pharmasset that have hepatitis C drugs that were just purchased with phase 1 data, and while they were completing phase 2 data for $2.5 billion and $11 billion. While they're not direct correlations, that both diseases, fatty liver disease and NASH, cause liver failure through scarring of the liver, and hepatitis C causes liver failure through scarring of the liver. There's a much bigger patient population in NASH. There's no competitors in NASH. So from a drug company executive's position, it's a very desirable metrics when you have 15 million patients, no competition, and a certain subset of those patients with the disease advanced far enough that they're going to be life-threatening. And you have a product that's made out of a carbohydrate that's safe and is inexpensive to make, unlike monoclonal antibodies, which can be $300 or more a dose to produce. You're talking about negligible prices here of $5 maybe. So we think the possibility exists we could have some initial inquiries with phase 1 data. Phase 2 is where we really expect to have the inquiries. And because in phase 2 we'll have biopsy data that will be able to demonstrate the reversal of the scar tissue. At phase 1 we'll have indications of improved liver function, but it's unlikely we're going to have the hardcore data on the reduction of scar tissue, which is the big deal in liver disease.
Thank you, Jim. I believe that will wrap up the Q&A. And again, I just want to say thank you on behalf of all those listening in and RedChip companies for taking the time to present at the RedChip Small-cap Virtual Conference.
We invite anybody to call the company with any questions that they have. The number's on the website. If they do that, if they could give us 3 or 4 times when they'd be available to call back, it might make it easier to facilitate hooking up with them and get their questions answered. I thank everybody for attending.
Thank you, Jim. That was Jim Czir. He is the Executive Chairman and Founder of Galectin Therapeutics. Galectin Therapeutics trades under the symbol GALT on the Nasdaq.
This video is not a recommendation or solicitation to purchase or sell any security, nor does it constitute investment advice. Visit redchip.com for full disclosures on RedChip clients.